Hi everyone, it's Nima with the BJYU Club. Welcome back to our YouTube channel. Today I have something a little different for you guys. We're going to be doing some microbiology practice rather than our usual organic chemistry. And we're going to be talking about the differences between gram-positive and gram-negative bacteria. So let's go ahead and get started. The first thing that I'm going to talk about is going to be the cell membrane, which is actually going to be the same exact in both gram-positive and gram-negative bacteria. And both of these guys contain a phospholipid bilayer. And what that is this layer depicted over here, actually for both sides. And it contains these yellow regions and these white regions. So the yellow regions are our phosphates. These parts are hydrophilic, they like polar molecules, and they like charged molecules. The white ends are the hydrocarbons or the lipid layer that is actually hydrophobic and does not like charged molecules, only likes nonpolar molecules. And what this does is it prevents certain molecules from entering or exiting the cell. In order for molecules to pass through this layer, they need to use certain proteins, which can be only on one side of the membrane or actually integrated into the entire cell membrane. And the two are actually depicted here. This one that you can see that is only on one side of the cell is known as a peripheral protein. And the one that actually goes through the entire cell membrane is called an integral protein. Moving on, we're going to be talking about the periplasmic space. And what this space represents for gram-positives is going to be the space between the peptidoglycan cell wall and the cell membrane. And in gram-negatives, it's going to be the space between the outer membrane and the cell membrane. This region actually has specific transport proteins or enzymes that allow for the cell to be able to take in certain nutrients. And the next thing that we're going to talk about is going to be the peptidoglycan cell wall. This cell wall is actually what keeps the cell from bursting open due to water pressure, as well as it is a defense mechanism for it and provides rigidity and structure to the cell. And one of the most important things that you need to remember is that the gram-positives have this really thick cell wall compared to our gram-negatives, which have a very thin cell wall. Now, these peptidoglycan cell walls actually are semi-permeable, meaning that certain molecules that are, large enough but not too large can actually pass through them, even though if they're charged or uncharged. So certain small peptides or certain ions or even sugars can pass through it easily compared to our cell membrane. Moving on, we're going to talk about another very important distinguishable characteristic between the two, and that is the outer membrane. And that is not present in the gram-positives, but rather is only present in the gram-negatives. And we have this outer membrane right over here. And the outer membrane is composed of two separate parts. We have this outer region, which is called the LPS or lipopolysaccharides. And these structures that you guys are seeing right now are actually just chains of polysaccharides that interact with the surface and the outside. And the inner side of this outer membrane is just a regular phospholipid bilayer, so similar to the one that is in the cell membrane. And similar to the cell membrane, this outer membrane also has proteins that allow for transfer of specific nutrients. And these are actually called porins. So these are also semi-selective, so they only allow for passage of certain things, but they're not as selective as the cell membrane integral proteins. So things like smaller molecules or medium-sized molecules can pass through it as long as they are uncharged. And the next thing that we're going to be talking about now is going to be the cellular connection. And what I mean by that is what keeps these certain regions connected to the cell membrane. And in gram-positives, these are called teichoic acids. And it's not actually in this depiction, but I can show it to you guys. So what teichoic acids do is they connect the peptidoglycan cell wall to the cell membrane. So if they were present in this picture, they would look something like this. They will start off from the cell membrane and they would connect to the peptidoglycan cell wall like that, where they covalently bind physically to the carbohydrates or the peptides that are in that peptidoglycan cell wall. And when it comes to gram negatives, they don't have this for the cell membrane and the peptidoglycan cell wall. Rather, they have another thing called lipoproteins. And what these do is they actually anchor the outer membrane to the peptidoglycan cell wall. And they're actually depicted here in this picture, right over here. And as the name suggests, it's just proteins that have certain lipids inside of them, lipoproteins. Moving on, we're going to talk about how resistant these guys are to antibiotics. And since most antibiotics target the peptidoglycan cell wall, the gram positives are more sensitive to these antibiotics because they don't have a outer membrane that could actually protect them from these antibiotics. So the drugs that target these enzymes in the cell wall could just reach their targets and affect it, causing the cell to burst due to not having this cell wall. But with gram negatives, they have this outer membrane right over here. So these drugs could not effectively reach these enzymes in the cell wall. So these guys are actually less sensitive to antibiotics. And lastly, what we're going to be talking about is going to be how differently they stain when we're doing a gram stain in a lab. And this is like a perfect way to distinguish between the two. If you guys have a specific sample from an environment and you want to see like if it's gram positive or gram negative, this is the way you would know. And gram positives stain purple and gram negatives stain pink. And I have some original photos for you guys so you can kind of distinguish between the two. On the left hand side, you can see that I have these coccus shaped bacteria that are staining purple. This is because these guys are gram positive. And on the right hand side, we have some pink bacterial cells that are bacillus shaped or rod shaped, and these are gram negatives. So that's all I have for you guys today. Please let me know if you have any questions in the comments. As I mentioned before, I'll be happy to help you guys. And yeah, I'll see you guys in the next video. Thank you.